So what are lipids? Lipids are oily or fatty substances that play three physiological roles. 1. In adipose cells, triglycerides, known as fats, store energy. 2. In cellular membranes, phospholipids constitute a barrier between intracellular and extracellular environments. 3. Cholesterol is a special lipid that serves as a building block for steroid hormones. The main characteristic of a lipid is hydrophobicity, which is water fearing. Water is polar. Polar substances dissolve well, which are known as hydrophilic. And CC, carbon-carbon, and carbon-hydrogen bonds are nonpolar and will not dissolve well in water. So sugar dissolves well as cooking oil floats on top. So hydrophobic means lipophilic, and hydrophilic means lipophobic. So fatty acid structure. These are composed of long unsubstituted alkanes with carboxylic acid at the end of each chain. These are typically 14 to 18 carbons long, and due to the fact that these chains are synthesize two carbons at a time from acetate, only even numbered fatty acids are made in the human cells. So saturated means fatty acids with no carbon double bonds. And the carbons are saturated with hydrogens because we need a maximum number of bonds. And then unsaturated means fatty acids that have one or more double bonds in the tail, and these are always cis or Z. So we're going to draw out these real quick. So this is going to be your saturated fatty acid. And now we'll look at the unsaturated fatty acid, which is going to have basically like a little kink. If we look here, we have your double bond. So how does the shape of an unsaturated fatty acid differ from that of a saturated fatty acid? So like I just previously stated, the unsaturated fatty acid is bent or kinked and forms a cis double bond. If fatty acids are mixed into water, how are they likely to associate with each other? The long hydrophobic chains will interact with each other to minimize contact with water, exposing the charged carboxyl group to aqueous environment. If we look here, we have a fatty acid micelle. The hydrophobic area with nonpolar tails, so this right here in the middle, is going to be lipophilic, and then out here the heads are going to be hydrophilic and polar, and these are lipophobic. So the force that drives the tails into the center of the micelle is called hydrophobic interaction. Water molecules must form an orderly salvation shell around each hydrophobic substance, and H2O has a dipole and likes to be able to share its charges with other polar molecules. The salvation shell, this allows for the most water-water interaction and the least water-lipid interaction. The problem with this is during formation of salvation shell, there's an increase in order, thus a decrease in entropy, and is unfavorable due to the second law of thermodynamics. So how does soap help remove grease from your hands? Grease is hydrophobic, lipophilic, and not soluble in water. Therefore, washing hands with soap causes micelles to form around the grease particles. Now we'll look at triacylglycerols, TG. And the technical name for fat is triacylglycerol or triglyceride. And these are storage form of fatty acids. The storage form of fatty acid is fat. So these are composed of three fatty acids esterified to a glycerol molecule. Glycerol is the 3-carbon triol HOCH2 attached to CHOH attached to CH2OH. And these have 3 hydroxyl groups that can be esterified to fatty acids. This is necessary to store fatty acids in relatively inert form of fat because free fatty acids are reactive chemicals. So triacylglycerol, this reaction is similar to esters. It's a base catalyzed hydrolysis. Soaps, sodium salts of fatty acids, RCOO and Na+. These are amphiphatic, and that means amphiphatic means hydrophobic and hydrophilic regions. So it's going to have hydrophobic and hydrophilic regions. So soap is produced by base catalyzed hydrolysis of triglycerides from animal fat and the fatty acids and salts. And saponification is the reaction that this goes through. So we're going to have your triacylglycerol, and then we throw some sodium hydroxide in there, and then this is going to form glycerol and then 3 fatty acids. And then lipases are going to be the enzymes that hydrolyze fats. So where are triacylglycerols stored? Fat cells as energy sources. Why are fats more efficient energy storage molecules than carbohydrates? And this is going to be due to packaging and energy content. So first we look at packaging. Packaging is the hydrophobicity, allows fats to pack close together. Carbohydrates carry a great amount of water of solvation, and the amount of carbon per unit area or unit weight is much greater in a fat droplet than in a dissolved sugar. So when you want to think about this, the packaging, think about storing sugars of dry powder in our bodies. It's not going to do much. It really won't. It just can't happen. And then for energy content, this is where fat has more energy carbon for carbon than a carbohydrate because fats are much more reduced. So energy metabolism begins with oxidation of foodstuff to release energy, and carbohydrates are more oxidized to start with, and if they're oxidized, they're gonna have they're not gonna release as much energy. So animals store most of energy as fat and store only a small amount of carbs as glycogen. Now we'll look at the lipid bilayer membranes. So these are membrane lipids. Phospholipids derived from diacylglycerol phosphate, so DGP. And phosphatidylcholine is a phospholipid formed by esterification of a choline molecule to the phosphate group. Phospholipids are detergents, so these are efficiently I can't talk right now. Phospholipids are detergents, efficiently solubilized oils while remaining highly water soluble. Detergents are like soaps, but stronger. And phospholipids minimize interactions with water by forming an orderly structure, lipid bilayer. So hydrophobic interactions drive formation of lipid bilayer and stabilized by Van der Waals and Waals forces. So would saturated or an unsaturated fatty acid residue have more Van der Waals interactions with neighboring alkyl chains in a bilayer membrane? Phospholipids composed of saturated fatty acids make membrane more solid because they aren't bent. Unsaturation has double bonds that in phospholipid fatty acids will increase membrane fluidity. And this prevents membrane from solidifying by disrupting orderly packaging of hydrophobic tails. So the right amount of fluidity is essential for functioning, and this decreasing length of fatty acid tails also increases fluidity. Cholesterol is the third important fluidity modulator. Low temperature increases fluidity in the same way as kinks and fatty acid tails, and this is known as the membrane antifreeze. High temperature of cholesterol reduces membrane fluidity. And the three main determinants of fluidity are going to be the degree of saturation, the tail length, and the amount of cholesterol. And the plasma bilayer surrounding cells, these are impermeable to charged particles such as Na, sodium. Protein gateways such as ion channels are required for ions to enter and exit the cell. Proteins that are integrated into membranes also transmit signals from outside of the cell into the interior. An example is certain hormones, peptides, can't pass through cell membrane due to charged nature. So protein receptors in cell membranes bind to hormones and transmit a signal into the cell in a second messenger cascade. Now we look at terpenes. These are member of broad class of compounds built from isoprene units, which has 5 carbons and 8 hydrogens. Terpenes are linear or cyclic, classified by the amount of isoprene units they have. So limonene, these are known as monoterpenes, and then we have like humulene. These are sesquiterpenes, which has 3 isoprene units. Monoterpenes have 2 isoprene units. Diterpenes have 4 isoprene units, and then so on. Squalene is very important. This is a triterpene. This is an important compound. It's biosynthetically utilized in the manufacture of steroids and is found in humans' earwax. So a terpene is a simple hydrocarbon, and terpenoids are functionalized terpenoids. These are This is an example of vitamin A. So what are steroids? Steroids are hydrophobicity. They have hydrophobicity, and these are similar to fats. They have a unique structure. They have a basic tetracyclic ring based on cholesterol. And these are polycyclic amphipathic. And the steroid cholesterol, this is an important component of the lipid bilayer. And these are due to diet and synthesized by the liver. These are carried in blood and packaged with fats and proteins known as lipoproteins. The type of lipoproteins are known as plaque, and this is the buildup of cholesterol, and this can cause atherosclerotic vascular disease. Steroid hormones are made from cholesterol, and the two that we're going to look at are testosterone, which is androgen, and this is the male sex hormone, and estradiol, which is estrogen, and this is the female sex hormone. So no receptors for steroid hormones, and there's no receptors for steroid hormones on the surface of cells. Steroids are highly hydrophobic, and they are able to diffuse light through lipid bilayer into cytoplasm, and they're located inside the cells. Peptide hormones are a little bit different. These are insulin, and an example is insulin, and this exert effects by binding to receptors at the cell surface, as a steroid hormone, such as estrogen, diffuses into the cells to find receptors. Phosphorus-containing compounds. These are inorganic and non-carbons, and there's no carbons. They have potential to donate protons, and the Kas for 3 acid dissociation equilibria are 2.1, 7.2, and 12.4. At physiological pH, phosphoric acid is dissociated and in anionic form. So phosphate is an orthophosphate. Two orthophosphate bounded via anhydride linkage form pyrophosphate, which is POP, pyrophosphate. And this is highly energy phosphate bond, and the hydrolysis of pyrophosphate is thermodynamically favorable. The three reasons phosphate anhydride bonds store so much energy is phosphates are linked and negatively charges repel. Orthophosphate has resonance forms and many resonance forms and lower free energy than linked phosphates. And the orthophosphate has more favorable interactions with biological solvent water than linked phosphates. So linked phosphates act like compressed springs, waiting to fly open and provide energy for an enzyme to catalyze a reaction. And looking at nucleotides, these are the building blocks of nucleic acids, RNA, and DNA. And these contains a ribose sugar, a purine, and a pyrimidine base joined to carbon number 1 of ribose ring, and 1, 2, or 3 phosphate units joined to carbon of the ribose ring, carbon 5 of the ribose ring. Nucleotide adenosine triphosphate. This is the central role in cellular metabolism in addition to being RNA precursor. So ATP is a universal short term of energy storage molecule. And this energy is extracted from oxidation of foodstuff and is stored in phosphoanhydride bonds of ATP.